All right, well, I'd like to welcome everyone to today's edition of the Polyploidy Webinar. Up first today is Dr. Anthony Baniaga. Anthony grew up in San Diego, California, and did his undergrad at Cal Poly San Luis Obispo, where under the mentorship of Dr. David Keel, he found his knack and love for field botany. After working as a field botanist for the United States Forest Service for several years in Northeastern California, he joined the EEB department at the University of Arizona, where he completed his PhD in 2019, working on Selaginella in my lab last year. And he's currently a Donnelly postdoctoral fellow at Yale University, sponsored by Michael Donahue and Erica Edwards. And today he's going to tell us about his work working on taking the glories of polyploidy and cytotype variation to Viburnum, and tell us about his work investigating mechanisms of cytotype coexistence of the Viburnum dentatum complex on coastal islands off the Northeastern US. With that, I'm going to let Anthony take it away.
Okay, well, thank you, Mike, and thank you all for attending. Hope everyone's well. I just want to apologize that there might be a beeping in my background, and it's because I'm in this growth room antechamber, and it's the only place I can get really solid internet and not wear a mask. Okay, anyways, an alternate name for this talk might be Natural History of Cytotype Coexistence as well. Okay, so we all know that polyploidy, is this gonna work? We all know that polyploidy is really important, right? We have intimate relationships with all of these polyploid species, but it's also really important for natural systems. So speciation by polyploidy is a really common process in vascular plants. Roughly 15% of speciation events in angiosperms and 30% in ferns and lycophytes are associated with a shift to a higher ploidy level. And we have a lot of reasons to kind of be excited about speciation by polyploidy. And that's because, as I'll outline, one of them being these physiological consequences of duplicating the genome. And so when we duplicate the genome, the nucleus and the amount of DNA increases, and that causes an allometric increase in the size of those cells. And we've known for a long time, reviewed in Darlington since the early 30s, that when we do this when we artificially increase the ploidy of moss gametophytes, we get a decrease in the cell number, and this corresponds to an increase in the cell, an increase in the cell volume. Hypothetically, this might scale up to physiological effects, as we can see these global trait correlations between increasing the genome size and the positive relationship with guard cell length of the stomata. And undoubtedly, these interact with evolutionary genetic processes, and whether you're an autopolyploid and an allopolyploid. But I think this is a really great time to study polyploid biology because, as Donald Levin outlines, in this time of biodiversity crisis, and in the next 100 years, most of the speciation events that might occur are polyploidy, and that's because polyploidy speciation can happen in less than 100 years. And we actually have current day examples of this, including Tragopogon up there in the Northwestern US. Okay, so for the outline of the talk, I'm going to first talk about my work on the Viburnum dentatum species complex, talk about some really cool findings of coexistence of Viburnum venosum and Viburnum recognitum, go on to talk about my studies on ecophysiological differences that might occur between the two, and then end with some notes about the reproductive ecology and really the role of disturbance in this system. So for those of you that are not aware, Viburnum dentatum is this really common shrub found in these mesic habitats all over the East. And so it does cross the Mississippi River, kind of in Iowa, Missouri, Arkansas, Louisiana, and even gets as far west as East Texas, but it's found all over the East, even into Canada. And it's this deciduous shrub with these broad leaves and these shiny blue fruits. And it's been this really tough taxonomic complex for a lot of systematists to circumscribe and treat ever since Linnaeus kind of outlined it and described it in the mid-1700s. So this is kind of a chart showing you all the taxonomic history of all of these great systematists have tried to outline and describe the diversity in this complex. The blue and the red kind of trace these widely distributed taxa these really distinct morphotypes. Part of the reason why this is really difficult to understand is because there's a lot of complex variation going on within individuals in the field. And one of those is Viburnum dentatum has this seasonal heteroblasty. So as the bud bursts and as those leaves develop along the shoot axis, they go from taking on these cordate leaf shapes here at the base to these more elliptical shapes. And depending upon when these things were collected and who collected them, you can get widely different characters to describe what you find on those specimens. Layered on top of that is this really strong latitudinal variation in leaf form. So specimens that are found around Florida have fewer teeth, and those teeth are not as well defined as if you were to go and collect something in the dentatum complex up in Maine. Finally, layered on top of that, we have a whole bunch of hair characters that range from glabrous to simple to stellate. Okay, so currently, Bruce Story and Alan Weekly, working out of the University of North Carolina, have very well defined kind of 7 currently recognized taxa. And as you might see from some of these names, such as Alabamensi and Carolinianum, these taxa kind of have geographic affinities, but also they have possibly genetic structure. And that was one of the things that I was brought here to Yale to understand. And that's because of work that was done in the 50s, starting with Egolf, counting chromosomes. And what he found was that Viburnum dentatum has a base 2N size of 36, while this described variety demii found in the Midwest. All of the accessions that he sampled had 2N equals 72. And then this once recognized taxon Viburnum dentatum variety pubescens has both 2N equals 36 and 2N equals 72. And so over the piggybacking on some work that was done by a graduate student, Beth Spriggs, over the last year, I've been trying to go and visit these sites and understand the geographic distribution of these ploidal variants in the field and collect them and bring them back. But for the talk today, I'm really going to focus on some of this really exciting work that we've found up here in the Northeast. And that's where we started, and it's been really tough to leave because of the cool stuff. So we were interested in visiting this area because there's this regional endemic called Viburnum venosum. And unlike the widespread Viburnum recognitum that occurs here up in the Northeast, these things are very different, and they have these very big, thick leaves. And so Michael Donahue was like, you know, before you really go out all over the US, you should really focus on what's going on up here, closer to home. And so that's where I started, and I haven't really left that area. So searching through floras and herbarium specimens, Viburnum venosum is really only found from this geographic area called the Long Island Cape Cod coastal lowlands. And this is comprised of Long Island, Block Island, Martha's Vineyard, Nantucket, and Cape Cod. And besides kind of having this different maritime climate, these areas do have a geologic shared history. And that's because these areas are the edge of these terminal moraines that happened during the last series of glaciations in this area. So these glaciers reached their maximum in these areas and deposited really nice moraines and clay deposits. Here's a photo showing some of those moraines on Martha's Vineyard down there in the water. And also just right on the beach right next to them, here are some really interesting clay deposits. I've never seen this before, but it's kind of fun. And what I think is going on when I visit these sites in these natural, where Viburnum venosum occurs most naturally, is that there are these really extensive clay deposits that occur throughout the island, and those create these really marshy environments where water doesn't evaporate as quickly. And you find Viburnum kind of ringing the edge of these swampy depressions. However, you also find them integrating into these prunus woodlands, and you also find them on roadsides. And this is true across all of the islands I've seen, But one of the first things I found when we went out there to look for Viburnum venosum across all of these different sites is that it grows right next to the widespread Viburnum recognitum. And we didn't know this really going in. Looking at herbarium specimens before, there's no GPSs on any of these things, but they mentioned you could find that from a couple of the islands that both of them occurred there together. We were in no way expecting them to be growing right next to each other. And these are readily distinguished in the field. So Viburnum venosum holds its leaves kind of horizontally while Viburnum recognitum holds its leaves much more vertically. In addition, Viburnum venosum has these much larger leaves on average than Viburnum recognitum. And Viburnum venosum is covered in this really dense stellate pubescence, while Viburnum recognitum is mostly glabrous and does have simple hairs that are kind of clustered in the primary to secondary vein juncture. Here's kind of a close-up of those two growing together, Viburnum recognitum on the left, glabrous, Viburnum venosum on the right, with that dense stellate pubescence on most surfaces. And so I collected a lot of data on these things, herbarium specimens, silica gel, pickled leaves, to study them more in the lab. But we also collected stem cuttings and grew these things up in the growth chamber and also in the greenhouse. And one of the first things we did was chromosome counts. So this is really exciting because we confirmed that Viburnum recognitum 2n = 36. It's a tetraploid, while Viburnum venosum, we didn't know this before, 2n = 72, and it's an octoploid. You might be asking, well, how do you know this is a tetraploid and this is an octoploid? Well, Morgan Mogley, a lab mate of ours in the Edwards lab, did a really nice survey of genome size and chromosome number across the genus of Viburnum to reconstruct where those shifts in chromosome number occur throughout the phylogeny. And so we know that Viburnum recognitum is a tetraploid, Viburnum venosum is an octoploid. I've confirmed this by doing chromosome squashes across all of those different sites where Viburnum venosum occurs and where recognitum is growing right next to it. And across all those sites, Viburnum venosum, 2n = 72, Viburnum recognitum, 2n = 36. We've also confirmed with flow cytometry that Viburnum venosum has roughly twice the amount of Viburnum recognitum across all of those different sites. And so this is really exciting because this suggests that we have coexisting tetraploid and octoploid taxa across multiple different sites throughout this region. But we didn't really know, what the evolutionary history of Viburnum venosum and recognitum were. Is there one origin of Viburnum venosum? Is it closely related to recognitum? Are we looking at multiple origins across all of these different sites? We didn't really know. So a while ago, we built a rad plate, and I've just started to infer that. And there's a lot of stuff going on because this is in the context of the entire Viburnum dentatum complex. But I want to draw your attention to this here, what I'm trying to show you right with my cursor. So we have pretty good evidence that Viburnum venosum is a single origin. Across all of those samples and all of the sites where it occurs, they all cluster together. And these are not really closely related to Viburnum recognitum up here. And we also have this interesting collection of Viburnum dentatum from North Carolina between recognitum, between venosum, of dentatum between venosum and recognitum. Okay, so that's all I'm going to talk about, right? That another story going on there completely. But I just want to summarize that we have really good evidence of coexisting tetraploid and octoploid taxa across all of these multiple sites, evidence for a single origin of the regional endemic Viburnum venosum, and it's not closely related to its widespread relative Viburnum recognitum. And so, walking around in these areas where both of these taxa occur and thinking about kind of my training as a polyploid biologist, thinking about minority cytotypes, how can we have these two taxa occupying the same place. And we thought that the best place to start to look was looking at ecophysiological differences. And yes. And we were thinking about this really in the context of some of these really interesting papers describing these nucleotypic effects and how they might scale up to metabolic effects. And the way this kind of line of reasoning occurs is that as you increase the genome size across all vascular plants that were sampled, yes, you do get increases in the guard cell length. This corresponds to a decrease in the number of stomata per area. This is also associated with larger genome size taxa having lower vein densities. And when you combine these three elements together, the hypothesis is that larger genome size taxa should have lower stomatal conductance than smaller genome size taxa. Scaling up, global traits correlations suggest that taxa with larger genome sizes should have lower maximum rates of photosynthetic gas exchange than taxa with smaller genome sizes. And so we thought, yeah, this is some compelling trait correlations. Let's investigate this where we have these very obvious differences in genome size. These taxa occur right next to each other in the field, and we've grown these guys up in common gardens. We would probably expect to see a difference if it doesn't indeed occur. And so when we started investigating this, yes, there are some pretty strong relationships between genome size or ploidy, however you might want to say it, and guard cell length, and also those larger, the octoploids have lower stomatal densities. And so we decided to scale this up and start looking at those maximum rates of photosynthetic gas exchange, but also performing light response curves of these guys, initially starting in the greenhouse because this was kind of during COVID times when we couldn't really go many places. And so we sampled 3 to 4 leaves per accession, and really 16 accessions of Viburnum recognitum and venosum that were collected across the range where these guys occur naturally, and we're growing right next to each other in the field and also right next to each other in these random arrays throughout the greenhouse. And so what did we find? Oh, what did we predict? So what did we predict? Well, we would predict because that Viburnum recognitum would have higher stomatal conductance and higher maximum photosynthetic rates. What did we find? Well, across light intensities, we find that on average, Viburnum venosum, the octoploid with that much larger genome size, has higher rates, higher photosynthetic rates across light intensities. And really no difference in maximum stomatal conductance across all of these leaves that we've sampled. When you take into account kind of the respiration differences, contrary to the prediction, we actually see a significantly higher difference in that maximum rate of photosynthetic gas exchange, with the Viburnum venosum having slightly higher but significant rates of maximum photosynthetic gas exchange. And really, after doing hundreds and hundreds of these measurements, I'm not sure there's a functional difference at all between having a mean of 13.17 and a mean of 14.72. That's within the amount of variation that you might find on a plant, and I'm not, in no way would I suggest that these are functional differences, at least measured at the leaf level. And so this really got me kind of thinking, okay, well, they're not in the greenhouse. I have the opportunity, let's go see what's happening in the field. And so we were able to go and measure the exact same individuals on Martha's Vineyard that we had measured in the greenhouse. And what we did is, across 3 sites, we measured 3 to 4 leaves per accession, 11 to 12 accessions of both of those different taxa. Okay, what do we find? Well, similarly to the greenhouse, those taxa performed similarly in the field, where Viburnum venosum had slightly higher rates of photosynthetic gas exchange across light intensities, although those rates are lower than what we found in the greenhouse. And so similarly, if we look at the metabolic differences between these two different taxa that might describe where they live, there's really no difference that convinces me that there is a functional difference between those two species. And this was kind of frustrating, and we're still kind of picking apart some of those finer anatomical details. And it's frustrating to kind of think that like, we didn't capture any strong functional differences between these two taxa that grow right next to each other. There must be some level of assortative mating going on. And as this photo kind of shows, yes, there is strong differences in flowering time between these two taxa across these islands. So Viburnum recognitum flowers in early to mid-July and has peak flowering around 7 to 10 days before Viburnum venosum, which has peak flowering in late June to early July. Here's kind of an interesting photo from Block Island showing it on the flip side, where Viburnum recognitum is mostly done flowering and Viburnum venosum is just about to pop those floral buds and start flowering. And so based on some of these observations, we think that if there really is any potential gene flow going on or any type of process that could occur in the field, it would occur from Viburnum venosum going to Viburnum recognitum. But hunt as we may, and I've searched across all of the islands, we have never found anything that looks like an intermediate individual. So in some way, these prezygotic isolating mechanisms must be fairly strong. And I'm following up on this right now by doing vernalizing these plants right now in the greenhouse and getting them all to flower at once, so that come a few months, we can do some really nice controlled crosses and see what's going on in a much more quantifiable way. At the same time, I've been kind of thinking like, okay, how is this possible? Okay, we have these two different taxa that are probably a result of secondary contact. They grow in high abundances, probably isolated pretty strongly, prezygotically. What's going on? And so I started reading a lot of historical floras from this region, and I realized that what this view that we see right here is really a really recent association of these plants. If we were to step back over 100 years ago, for the last several hundred years, all of these islands look like these very vast prairies, very little woody vegetation at all. And even you can go and find that Henry David Thoreau wrote about this, that when he walked across Nantucket, he found very few trees except those found around houses. The entire landscape looked like a prairie. And this photograph kind of encapsulates that. And Peter Dunwoody has done some really interesting work looking at how these historical changes have affected these plant communities on these islands. And so here's a palynological record from Donut Pond in Nantucket showing that, since European colonization of these islands, they started cutting down these trees relatively rapidly. And we actually have historical records of people complaining about people cutting down the trees in these groves and people getting punished from them. And so after about 300 years, these pretty much the oak-dominated landscapes, in conjunction with massive amounts of sheep and livestock grazing, turn these areas into these really vast prairies devoid of lots of woody vegetation. And what must have happened out in these landscapes is that Viburnum dentatum, represented by recognitum venosum, must have been occurring out there at very low densities. And after all of this intense disturbance over multiple centuries, we have this really rapid regrowth. And so here's a repeat photo of this exact same spot done by Peter Dunwoody in 1986. Everything in the foreground obscures everything in the background. And what we have is this massive regrowth of cedar, oak, and what's coming up here in the foreground? Viburnum recognitum and Viburnum venosum. And so thinking about like how it is that we can have these two different species that are really functionally equivalent, although there might be some subtle variations going on there, how it is that they can coexist. I think that there's a really strong role of disturbance in facilitating some open habitat for these guys. And we're seeing the regrowth after centuries of disturbance. And it will be interesting to see if there is any type of replacement of one species versus the other. This is kind of a nice system to kind of explore those because they occur across all of these multiple different sites. And with that, thanks for listening. A lot of people to thank at Yale for field assistance and for all of the research permits that allowed us to collect and study these plants all across the islands. So thanks for listening. Happy to discuss any information. Thanks again.
All right, well, thank you, Anthony. Yeah, we have time for a couple of questions, and it looks like Brittany is already jumped in there. Let her fire it up.
Hi, Anthony. First off, fantastic talk, really cool stuff. Just to make sure that I have this right, is it the venosum that's the octoploid and the recognitum that's the tetraploid?
Yeah, I'm sorry that wasn't clear. So Viburnum venosum is
I was just making sure.
Yeah, Viburnum venosum is the octoploid, recognitum the tetraploid.
Okay, so the two questions that I have then are, first, do you find 2n equals 54 either in these sympatric zones or in recognitum only populations?
Yeah, so it's a bit of a complicated question because no, we don't find any intermediate looking individuals. And of the individuals I've surveyed both in genome size and through squashes, we've never found a 2n equals 54 individual. However, Egolf, in his more extensive chromosome survey, at the top, he discussed this 2n equals 54 individual that was counted by someone else in England. And it's kind of this unclear because we don't know the source of that accession, whether that was a miscount or whether that really was something legit that was a hexaploid. And so we don't see it in the field and unsure whether it occurs, but these controlled crosses that I'm going to be doing, we might be able to generate those.
So next question, and this will be my last one. You mentioned about 2/3 through your talk, expecting to find, if you're having sort of introgression happening between the two species, finding venosum, interbreeding with recognitum more than the other way around. And I was just curious why you would expect that pattern, given that recognitum is the one that has the lower genome size. Because I would expect it to go the other way around.
So let me understand. So your question is, why do we think that the way that gene flow occurs is from venosum to recognitum?
Right.
Yeah, and so that's really based off of just observations in the field.
Okay.
And the phenological differences and the way that I've watched the stigmas mature and the pollen mature. So the pollen matures probably right after the buds.
Okay.
And then the stigmas mature several days later.
Okay, that makes sense. Because I was thinking about it just from a, typically with interploid crosses, you tend to find higher survivorship when the mother is the higher ploidal level. So, but yeah, that makes sense if you're looking at it, if phenology is not on your side there, that makes sense. Cool, thank you.
Yeah, cool questions, thanks.
All right, looks like the next question there is from Doug Soltis.
Do you want me to read it? Or
Yeah, go ahead. It feels a little bit like, like a more normal.
Really interesting talk. I appreciate it. And I love to see somebody who can actually make chromosome squashes. Oh my gosh, a dying art form. But so what are the parents of the two polyploids? And, to me, that might underlie some of your features, physiological morphology, flowering time, et cetera, that could be critical to moving forward. Thanks a lot.
Yeah, thanks, Doug, for that note. I agree with you, kind of understanding what the origin of Viburnum venosum is still quite elusive. We're not sure whether it's an autopolyploid or an allopolyploid, and really where it really clusters with other members of the dentatum complex. I tried to skip over it, but I'll show you it right here. Here's the phylogeny. What's really troubling to us is that you have these nested tetraploids within octoploids. And so what that suggests to me is that there probably is some type of history of hybridization in there. And it would be nice to know in a better way what the history of the parents of venosum is. I'll lastly say about that is looking at the phenotypes, or just looking at the morphology of venosum and then other members of the dentatum complex that occur in the mid-Atlantic coastal plain, it seems fairly, it seems like quite plausible that venosum is a segregate of these octoploids that occur kind of in New Jersey, Virginia, Maryland, that possibly during the last 5,000 years kind of made its way up into the Northeast and kind of got stuck there. That's one possible scenario, but I agree with your comment. Thanks.
So do plastid markers give you any insights as to the maternal parentage? I like your hypothesis, by the way. I mean, it could be some glacial activity that something that's further south might be apparent, but I just wondered if the plastid markers gave any insights here.
Yeah, so we don't have plastid markers that were sampled at this scale across so many different individuals. We haven't really looked yet to see if we could recover any of that from the RAD data. Previous publications, though, on Viburnum and the dentatum complex suggest that it follows this similar topology where you have scabrellum coming out as the earliest diverging member of this group, followed by dentatum, and then followed by recognitum. And so the plastid phylogeny, at least at that larger scale level, shows something similar to this topology.
All right, well, thank you. I think there's a couple more questions, but we'll take those at the end after Jeanette's talk. We'll have plenty of time then so that we can get the next talk going. But thank you, Anthony, for a great talk. And if anybody else has any other questions for Anthony, save those and we'll come back to those to circle around again at the end. All right, we'll let Jeannette go ahead and get her screen sharing. All right, so our next speaker today is Dr. Jeannette Whitton. Jeannette did her undergrad at McGill and her PhD at the University of Connecticut with Bob Jansen and Ken Holsinger. She then spent a year at the University of Texas before joining Lauren Reesberg's lab as a postdoc and then moving on to UBC, as she says, many years ago. After working on Crepus during her PhD, she followed Stebbins' advice and started working on the simpler Townsendia apomictic system, which I'm sure is much simpler. We're going to hear a little bit about that today. Jeannette is currently the director of the UBC Herbarium, the current vice president of the Canadian Society for Ecology and Evolution, and she's also in her second year of a 3-year term as the group chair for the Ecology and Evolution Discovery Grant Panel at INSTRC. And Jeannette is here today to on Thanksgiving Day in Canada, which we all realized a little bit too late, and so I'm very thankful that she's here to tell us about her work today on Townsendia, which she's going to tell us about the ecological and reproductive contributions to range limits of diploid and polyploid Townsendia hookeri. And with that, I'll let Jeannette take it away. Thank you.
Thanks, Mike. I'm really excited to be here, holiday notwithstanding. I think I have a tendency to forget holidays. I once invited Pam Soltis up to UBC to give a seminar, and it turned out it was the Easter long weekend. I don't know. So I'm not a very observant person, I guess. But I'm excited to talk to you today about this work, which I think is really finally starting to come together and gel into something resembling a story. And that's largely through the efforts of Evan Hirsch, who finished his PhD just this past June. Evan is a little bit more observant than I am, and so even though we talked about who would present, he's off celebrating Canadian Thanksgiving. Okay, so I'm just going to launch in and tell you some things about Townsendia, and specifically about Townsendia hookeri, the species that I'm going to focus on today. So this is a species of middle elevations. It occurs on the Front Range in Colorado and Wyoming and extends mostly up the east slope of the Rockies to the southern Canadian Rockies. And there's also a disjunction up in Yukon. There's a small set of populations up there that we won't talk about for the rest of today. It's called Easter daisy probably because it flowers soon after the snow melt. In fact, the plants produce buds in the fall, and as soon as the snow comes off them, they'll flower, which leads them to sometimes flower in November if they get an early snow and then a thaw. But I think that flowering is inconsequential with respect to seed set. They're impressively long-lived perennials, and they're impressively tough. We've had plants in seed packets on the bench and decided to, what the heck, why not stick them in a pot and water them up and see if they grow, and they come back, which gives another possibility to why they might be called Easter daisies. We know that the apomictic polyploids in this group have arisen at least 3 or 4 times from sexual diploids. So in this case, all of the polyploids that we know of are apomictic, and all of the diploids are sexual outcrossers. And we can talk about the evidence for that if you have questions. The apomicts are diplosperous, and so what that means is that the cell that should have undergone meiosis and given rise to a haploid egg has an arrested meiosis, a restitutional meiosis, and gives rise to a diploid megaspore mother cell that then starts to divide and form an embryo. These apomicts don't require pollen to initiate endosperm formation or trigger anything, but they do produce an average of about 25% viable pollen, presumably because they just haven't gotten rid of it yet, haven't accumulated enough mutations to stop doing that. There is an odd population in Yukon that we know of that doesn't produce anthers at all. And so just to emphasize here, this is a sort of a bimodal distribution. We're looking at pollen stainability on the y-axis and pollen diameter on the x, and there's really 2 clouds of points. There's those 2 little points down there that suggest the possibility of something with small genome and low pollen stainability. We've never refound plants like that in our surveys. And so I don't know what to tell you about those at this point. Nonetheless, a little bit of spread notwithstanding, as I said before, everything that we know of as diploid is sexual, and everything that we know of as apomictic is triploid in the southern part of the range. And the plants show this classic pattern of what's called in the asexuality world geographical parthenogenesis. And that's a term that was coined by Albert Vandel in the 1920s. And it describes a pattern in which sexuals have a smaller and usually more southern range than their related asexuals. Now, Vandel described it as a pattern. He didn't really postulate why that pattern might exist, but there's been some speculation since then. And of course, if you don't work on asexuals and you work on polyploids, this pattern will perhaps ring some bells. We know that there are many polyploids that show these kinds of distribution patterns with differences in the distribution of cytotypes, even though there's still a lot of conversation happening about what the general pattern is. So here's some big questions. I always do this in my lectures. I say, what are the big questions in this area? And then I don't always answer them all, and so I'm warning you now that we're working towards answering these big questions. So the first is, why do sexuals and apomicts have different ranges? And so I tend to say sexuals and asexuals because that's the context in which I work, but we could just say diploids and triploids. Why do they have different ranges? And does asexuality contribute to that distribution, or does polyploidy contribute to that distribution? Or, and I'll say it this way, does speciation contribute to the distribution? So is the distribution really about the evolution of reproductive isolation, regardless of whether or not that's brought about by asexuality or polyploidy, for example? So why might there be different ranges? Well, we could hypothesize that there are different ecological tolerances of the cytotypes that might result in them having different ranges on the landscape. It's possible that the distribution could reflect dispersal advantage or reproductive assurance of asexuals. So the sexuals in this system are obligate outcrossers. So they require dispersal of compatible mates. They require pollination, for example. The asexuals require none of that. And then another question we might ask is, are there effects of precedence? So does who can establish an range depend on who is already there? So these are some of the maybe more mechanistic kinds of questions that we've set out to design experiments and studies to address. And so what I want to talk to you about today is the biggest experiment that I've ever been involved with. Okay, maybe that's not exactly true, but I think it's true for my lab anyhow. And that's this multiple reciprocal common garden experiment that was conducted over a 5-year period. It had some perks. This is one of our garden sites here out in northern Colorado. And you can see just down here in the corner, this is our garden plot. The benefit of working on small plants is that you don't need very large footprints. And over here in the corner, you can see an establishment plot, and I'll mention those as well in a sec. So first, I want to mention how we thought about the context of testing range limits, and we were motivated by this framework that Anna Hargreaves, Karen Samus, and Chris Eckert had published in 2014 that makes some predictions about range limits and how we might interpret results of transplant experiments. And so we're looking at a figure here from their paper, where we would have on the y-axis population growth. So this might be lambda, for example, and that dotted line across might represent a cutoff, lambda, greater than 1 above, less than 1 below, where population persistence would be possible if you were above 1 and not if you're below. And then on the x-axis, we have the position within the range. And so in this example here, this cartoon example, in the interior of the range, that point over on the left, the population is well able to persist as we move towards the range edge in that center point here. Population is having a little bit more trouble, and then beyond the range, the population growth rate falls below 1, and we expect that the species wouldn't be able to persist. So this is a context in which the range limit essentially represents a niche limit, so RL equal NL. That doesn't have to be the case. In this case, even though there's a declining population growth function beyond the edge of the range, even beyond the edge of the range, populations are able to persist because they have lambda greater than 1. So this kind of framing motivated the design of our experiment. And so, oh yeah, right. If you have a situation in which the data indicates that a population can persist where it currently is not, then the interpretation would be either that there is dispersal limitation. So that's the sort of go-to first thing that you would learn as an interpretation. They're not there because they haven't gotten there. But there are a couple of other possibilities, one being design issues. So for example, if you've done experimental manipulations beyond the range edge, for example, to reduce competition, then that design could lead you to find something that wouldn't actually occur under more unmanipulated conditions. And there can also be unmeasured effects, so things that you were not able to consider, and that could include things like you happen to get a better year than some condition that could be limiting to persistence. So in our case, what we did was we thought about these 2 ranges and did reciprocal range limit experiments, moving the sexuals out of their range north and moving the asexuals out of their range to the south. And that resulted in a design in which we had 8 common gardens, 2 northern gardens that are in sort of the allopatric range of the apomicts, where there are no sexuals even geographically overlapping. two sets of two gardens in this overlap range, which are either in conditions where sexuals grow or in conditions where asexuals grow, but in this broad range of overlap. And then two southern sexual sites where essentially the sexuals are in allopatry, where there's no known overlapping apomicts. And we planted seedlings from 12 source populations into each of these gardens. And so this is just a little bit more, more detail, but just to clarify, on the 12 sets of populations that we used as sources for this experiment, we had 3 that were asexuals from the allopatric range, 3 that were asexuals from the overlap range, 3 that were sexuals from the overlap range, and 3 that were allopatric sexuals from the southern part of the range. Happy to answer any questions about design when we get a chance. So we grew up the seedlings at UBC, and I would say that this kind of experiment I wouldn't dare to try under current conditions. We were able to get these across the border as plants. We had all the permits, but that isn't enough anymore these days, have to say. And we put them into gardens in 2014. And I can say we when I say we put them into gardens because I was there that year, but I didn't return until the harvest year. So Evan and field assistants gathered data in the interim on establishment, survival, and number of flower buds, and a few additional traits, but these are the main traits that we measured. I'll mention here these establishment gardens. We knew that if we put seeds out in the field, we would get mostly zeros. Just about everybody would die, and that's a lot of work to get zero data. But we knew planting 3-month-olds out there that we were risking not capturing that early life history stage. And so Evan planted a parallel seedling establishment experiment that he checked after 1 year. Well, he checked it after 2 years, but nothing germinated beyond 1 year. So here we go. Here's one of our big punchlines, and this is the seedling establishment data. And so a few graphs are going to be oriented in the same way here. We're looking at predicted establishment success. So these are the output of GLMs. And color-coded are the source regions for the plants. And then we're showing from south to north the garden location. So the sexual allopatric gardens on the far left, the asexual allopatric gardens on the far right, the overlap zone in between, sexual, asexual. And so what you can see here are these really strong garden effects and really not a whole lot else. And so establishment seems to be a function of local conditions. And I want to put a little caveat here in that this is a 1 year experiment. So this is what happened when we put seeds into the field in 2014 and came back and measured who was alive in 2015. And so that's going to be relevant for a point that I'm going to make in just a few minutes. So there's not a whole lot to say about the differences between cytotypes or reproductive types based on these data. So the rest of the data are pretty complicated. So because we have 5 years of data, we did that because we wanted to get something closer to lifetime fitness for a long-lived perennial. But that generates huge headaches in the analysis. And so we did aster modeling, or Evan did aster modeling. This is a real pain in the tuchus, I have to say. And so what this framework allows is to think about the fact that in order to produce flowers at the end of the 5th year, you have to have survived all of these other years. And so the model's aim is to incorporate that year-to-year survival and the year-to-year production of flower buds into the estimate of lifetime fitness. I'll say as well that the closest we could get to lifetime fitness was this number of flower buds. And that's because we had permit conditions that didn't allow us to let our plants go to seed. I'll talk a little bit more about that in a bit. Okay, so here's the results of those survival and flowering data. And you can see that it's not all site specific. It's not totally governed by site conditions as the establishment data were. And so what we can see here, for example, well, actually, let's make this a little bit easier. Let me make sure I know what's coming up next. Oh, okay. I'll skip back to that other slide, but here we're just looking at the sexual diploids and their graphs. So I've just taken away a couple of lines here. In the context of the range limits theory, so let's just remind ourselves that the two sites that are surrounded by the blue box are outside of the sexual range, close outside or far outside, but in both cases, they're not sites that sexuals currently occupy. And what you can see is that there's substantial ability to survive and reproduce beyond the current range. And so in a simple niche limits, range limits context, this sort of supports that interpretation that would go to dispersal limitations. So it looks as though sexuals are able to persist beyond their range, but they're not there. I'll also just point out that the sexuals from the allopatric zone, the southernmost sexuals, do best in the southernmost sexual gardens, and those gardens seem to have been pretty hard on most of our transplants. And I'll just go back to this here, showing all of the graphs. There's our local sexuals versus the everything else. I'm going to skip over this next slide and go to the asexuals. And again, we're looking at things that are outside their normal range, but now they're on the left. So these are the sexual locations, and these are the asexuals' performance in those gardens. And here we see what is more in line with something like niche limitation. Our predicted lifetime bud production, our measure of fitness here, declines outside of the range where apomicts currently occur. Now, we had these two sets of data, the establishment data and the survival and reproduction data, but they weren't fully compatible. And yet, it's really tantalizing to try to get those data to come together. And so we used a kind of a resampling approach. And the reason they're not fully compatible is that, of course, the seedlings that were in the garden are not the adults that we then track later on, right? And so we used a resampling approach to generate new data points by resampling, I think 1,000 times, combining the data together in an appropriately structured way to generate a measure that incorporates the 5 years of survival and bud production with the establishment data. And that's what I kept skipping over here. And that's these data here. And what you can see here is that we go back to something in most garden locations that is a lot more like the establishment data, where we don't see these as many big differences. For example, in the sexual gardens, we no longer see that separation of the local sexuals doing way better. And that kind of makes sense because relatively few of the seeds survive and establish. And so that drives the overall patterns that we see here. There's a little bit of separation in the overlap gardens here. It's the two populations, one sexual and one asexual, from the overlap region that seem to be pulling apart. But overall, we see these strong site-specific dynamics. And yeah, we're going to be curious to see how this goes through the review process. This paper hasn't been submitted yet, but we've been encouraged to combine the data as we've gone through, committee review and that sort of thing. But one of the challenges that I have with that is that we really have that 1 year of data for establishment. And those strong site-specific effects may represent sort of very localized conditions. When did the rain fall in that particular site? And so I'm hesitant to take this as our best estimate of the overall dynamics of the system. Again, I'd love to get some feedback on that from you. Okay, so I did want to add a couple of things. So we did measure a few environmental variables and a few traits that I won't talk about, but I want to point out a couple of things. These are a few climate variables from the garden sites, and the shapes that you see are 100 years of data for mean annual temperature and mean annual precipitation on the right, and the dots show you the years in which the garden experiment was conducted. And so even though we're on the right skew of these 100-year distributions, we're not way outside of them. And so, you know, that's comforting in that we didn't just measure something that's a complete anomaly with respect to the relatively recent historical past. We also measured vegetation cover at the garden sites. And this, we could kind of see this when we put the experiment in. We didn't do anything to manipulate the garden vegetation at the site. We just planted things in as close to intact as possible, as long as they weren't going to hurt us, like the certain plants were inclined to do. And you can see that there's much greater vegetation cover in the southernmost gardens, and so that may be part of what's limiting the persistence of non-local types there. But we didn't do a manipulative experiment, so this is correlational at this point. So what did we learn from the Common Garden experiment? Well, we learned that establishment success is dictated by local conditions, and I don't really know how that varies from year to year. Our inclination is to say that sexuals, whoops, that's not a sentence, that sexuals don't show niche limitation. We see the ability of sexuals to persist in the gardens that are well beyond their current range. On the other hand, apomicts perform poorly in the sexual range, and especially in the allopatric sexual range, in the southernmost range. So what's going on here? So one of the things that happens in these asexuals is, of course, that they benefit from dynamics related to Baker's law. As I mentioned at the beginning, they don't need a mate and they don't need pollinators. And so it's possible that the sexuals are establishment limited in the north. They could do fine if they could just get a foothold. And they can't get a foothold because they need to establish in big enough numbers to have mates, and that just hasn't happened. It could also be pollinator limitation, and I can say that I don't think that's the case now, because we definitely observed pollinators, and even though we weren't supposed to allow pollination to occur, sometimes when we got to the field, we could see that that seeds were actually starting to develop in our plots, in the sexuals. We know that apomicts have a dispersal advantage based on just being able to establish a population with a single seed, but we've also measured other dispersal traits related to the pappus, and apomicts seem to have a dispersal advantage there as well, although, you know, over these scales, that would have to play out over quite a long period of dynamics. But in any case, if what we're thinking about is establishment limitation, this is not an equilibrium distribution then, because we would presume then that it's a matter of time, that sexuals should be able to catch up, that there's nothing actually preventing them from establishing in these sites beyond the getting there parts. Of course, it's possible that there's experimental limitations. We worked in this particular framework at this particular time, and there are things that we might have done differently that could have led to a different outcome. Mainly, we didn't have these super extreme years, and the year before we started this experiment was one of those extreme years in which there were very late snowfalls that really hammered the plants and prevented us from getting the seed we needed for this experiment. So we know that we were fortunate in having conditions that were conducive to more plants persisting. So we have to think about that too. But another thing that we know about is asymmetric reproductive interactions, and those could be acting to prevent invasion of sexuals. And I'm just gonna take a look at the time here, and I'm kind of at time. I don't know when we started, Mike, my chunk.
You got a couple minutes, go ahead.
So I'll just, I'm referencing here some prior work that we've done, and I'll just summarize it super quickly, or simply, which is to say that we know that there's asymmetric reproductive isolation between apomicts and sexuals. The apomicts are in their little bubbles here, and if there's sexual pollen flitting about, they're not susceptible to fertilization. We see this because apomictic embryos are actually precocious and have already started developing by the time the flowers open. And so there is no minority cytotype cost for apomicts. Whether they're rare or common, they're good with having sexual pollen around them. On the other hand, sexuals do suffer from minority cytotype effects, and even potentially from majority cytotype effects. We don't know exactly at what point these costs become insignificant. Pollen from apomicts can sire hybrids with sexual moms, and we also see facilitated selfing with sexual moms when you apply apomictic pollen, so mentor effect selfing. And given that these are long-time outcrossers, we would expect that that would likely lead to inbreeding depression, though we haven't measured that. In either case, no matter what's going on, we see these as fitness costs on sexuals. And we've done experiments, field experiments on crossing, and I'll skip over this at this point, but I'll say that I'll show you the essentially the outcome of those experiments, and that's that where there's a cost, it's always on the sexuals. And so we know that these dynamics are at play, at least in the experimental context where we've done this work. And that's likely to be a factor, supposing that sexuals could establish in the north. Now that the apomicts are there, the barriers to overcome in establishing sexuals in that apomictic range are actually quite high. So back to these big questions super quickly. Why do they have different ranges? Well, it's a complex story, and I think it includes partially some ecological conditions limiting the apomicts, and potentially a more complicated story for why the sexuals are limited. Does asexuality contribute to the distribution? Yeah, I think so. And I think that's primarily through the dispersal advantage that asexuals have that lead them to have that colonization advantage. Does polyploidy contribute to the distribution? Well, to the extent that we see any kind of ecological differences, I think that's polyploidy in action, but it's really difficult to disentangle those in this system. And finally, does speciation, or the evolution of reproductive isolation, contribute to the distribution? Well, I think those asymmetric costs of coexistence are likely to be important. And so I will just stop there and say thank you, but I'll also say that we're looking to continue this work and expand this work as well into other systems. And if you're looking for opportunities, please get in touch.
All right, well, thank you, Jeannette, for a wonderful talk. As always, let's put our questions there in the chat box. And if you want to, please also fire up your audio and video. It's always nice to hear everybody ask their questions rather than reading them off the list. And I have a couple of questions, but I was going to hold off until give everybody else a chance. But I'm going to go ahead and ask one here real quick. So, so do you think? I was trying to sort of disentangle too from the experiments that you designed. If there was a role for competition to among the sexuals and asexuals to play a role in those plots, or if that was sort of excluded as a part of the experimental design.
It wasn't excluded, but it wasn't accounted for either. So our spacing was about 10 centimeters. We plant every 10 centimeters, which is higher than most of the densities that we see in natural populations. But also in natural populations, like the plants can get quite a bit bigger over many
right.
years. And so we didn't explicitly measure that. I think we have the capacity to dig in a little bit, but that's what I think the next logical experiment is to actually plan an experiment that combines the measuring of competitive interactions and reproductive interactions.
Right. Yeah. Yeah.
In the two gardens. So that's going to be the next big thing.
But I have another real quick question that follows it up, and sort of in this the spirit. How do you is it possible for sexuals to be converted to asexuals? Sort of thinking about sort of the series. Is it you know, the direction of the origin, I guess, of the apomicts, and do you see any asymmetries in that as well, or anything?
Yeah. So the data that we have on origins are from plastid DNA, old data that suggests multiple origins, like 3 to 4 origins. And those are pretty coincident with the analysis of the SNP data that Evan has in terms of the specific origins. But that's not exactly addressing the possibility that apomixis could move as a little virus through this kind of crossing.
Right. Yeah.
Apomix and sexuals. And the best I can say there is that in the we kept the offspring going from the crosses that Alicia had done. We kept them going in the growth chamber for a long time, a couple of years, and a few things flowered over that time, and none of them produced seed. So we didn't see it in this very small experiment, even in the polyploids that we kept growing. Yeah, we also have to remember that these are triploids crossing against diploids, and so we would expect that if that was to happen, we'd see tetraploids actually.
Right. Yeah.
And we've yet to find a tetraploid in the southern range.
Right.
So that too is sort of circumstantial evidence that that pathway is not super common.
Yeah, that's cool. Well, yeah, this is awesome. I have other questions, but I'll ask you some other time. I want to see if anybody else has any other questions here real quick. All right. Well, while we've got everybody here too, maybe Anthony could pop back on and answer a couple of those questions while other folks are typing things in. Let's see. So there were a couple of questions, and these are all, you know, such great talks to pair together. So maybe Anthony, there's a question from Rob Laporte that's still sitting there for your talk. Does it seem that there might be community equilibrium dynamics that explain the coexistence of those cytotypes?
Oh, I liked your talk, Jeannette.
Thanks.
I can ask you some questions about some of that work, maybe in an email.
Awesome.
I guess for Rob's really.
I think Rob's still here, right? If he wants to pop on. There we go. Anyway.
Sorry, I have to balancing a couple of things here. So, but yeah, I'd like to just follow up because I think there was a lot of parallels between the two talks. So thanks, Mike, for putting together a really great pair of talks. And Jeannette, that was really awesome work. And Anthony, it was really cool to see how your work's becoming really comprehensive now. It's really cool. I think that Jeannette addressed this a little bit in hers that I was curious about after seeing your talk is just, is there like not a long enough time axis to really know what the sort of competitive or community outcomes of these co-occurring polyploids might be in those northeastern islands? It seems like that could potentially be playing a role in the Townsendia as well. So I don't know what your thoughts are on that.
Yeah, I would see really similar parallels thinking about tracking the retreat of some of those glaciation events. Maybe Jeannette can.
Yeah, I mean, you know, most of the time in our populations, the nearest neighbor to a Townsendia is not a Townsendia. So does that represent the outcome of competition, or does that tell us that competition isn't super important in this system? And I don't know the answer to that. But if you think about it, the physical range of competition is really quite small, especially relative to the physical range of these reproductive interactions. And so there has been some modeling work in these sexual-asexual systems that suggests that competition would have to be kind of really quite strong. Resource competition would have to be really quite a bit stronger than we typically imagine for it to result in exclusion. On the other hand, these reproductive dynamics can be really slow acting, and so it can take a lot of generations. And so especially in a long-lived perennial system, which both, I think is relevant to Anthony's work as well, you would expect that if things manage to establish, and that's how that happens is a question in itself, then the exclusion of one or other form through these reproductive interactions would likely take a really long time. And so, like in Anthony, in your system, I wondered about the possibility of character displacement, and if you looked at the allopatric populations of the widespread polyploid, whether or not you see any differences in the phenology.
Yeah, I've been thinking about how to tackle some of those questions too. It's a great comment. One thing I can say is we started looking at phenological differences that occur in the more widespread tetraploid across its range compared to its flowering phenology on the islands. And I'm not sure what to make out of it because there's an interaction there with climate.
Mm-hmm.
And so I'm not really sure how to separate those things. But yeah, I agree.
Yeah, you'd have to do it in common garden, which would be a huge undertaking.
You have some of those viburnum in the greenhouse now that they're clonal, right? So if there's a genetic effect of those clones, you should see some of that in the greenhouse if you can get them to flowering. I don't know how many years it takes to get to flowering.
Yeah, they'll definitely start flowering after we start vernalizing them. And they have occasionally they do weird things in the greenhouse, and they have occasionally started to flower, but not in any real consistent way. But we do have kind of a good way to initiate them to all start flowering within the same kind of couple of weeks. Let's think about that, Rob. Thanks.
Yeah, I find both of these stories so fascinating, and it's really interesting to see the question still of the origins of these, different groups is still really, really relevant. It would be really fun just to plaster all of these with a ton of genomic data and with close relatives and try to figure out, how much of this is, the real history of sort of movement of the cytotypes across the landscape or competition and replacement. I'm sort of very curious about all of that now. From hearing both of these, yeah.
What our genetic data suggests, both the plastid data and the SNP data suggests that the triploids that are in Alberta and Saskatchewan and British Columbia and the Dakotas are actually more closely related to the Yukon sexuals and have come down, and actually their cytotype patterns match really nicely some work that's been done on bison, including fossil bison and sort of time calibrations about when the ice sheets were parting. And so my hypothesis that Townsendia is bison dispersed is still alive and well.
Lauren always says this about the sunflowers as well, right? I'm sure you've had that conversation with him. But it's absolutely true. All those achenes get in the fur and just take a ride, right? I mean, that seems very plausible. And with the way, and they had that corridor, right? In the Ice Age, that would have been, things could have come down along the front range, I guess, through Alberta, right? Yeah, that's amazing. I'd love to see that. It would be really fun to see that reconstructed further and other work. It's a really cool system. And I'm super excited about it. I have a ton of other questions, but a lot of them are actually kind of hit here in this with Rob's question to both of you. So yeah, well, I think that looks like it's all the questions for today. And I just want to thank. Does anybody else, real quick before we wrap it up, have any other questions for either Jeannette or Anthony? All right. Well, I want to thank again both Anthony and Jeannette for 2 great talks today. And I want to highlight our speakers next week, for the next webinar here in 2 weeks. We have Veronica Destilio, who will tell us about interdependence of polyploidy, unisexual flowers, and wind pollination in a ranunculate. And Juan Losada, who will speak on the vascular function of subtropical fruit trees with different ploidies. And so we'll hear some more fun work at that interface of ecophysiology and phylogenetics and diversity. With that, I thank everybody for showing up today, and look forward to hopefully seeing everybody here soon, sometime after COVID ends. We've been doing this now for 7 months. We'll keep this webinar going this year for sure, and I'll be in touch with some folks again as we wrap up. I know that we had talked about maybe doing a couple panel sessions towards the end of the semester, and so I'll be in touch with some people about maybe making a couple of these here this year, just some open discussion groups, which I think would be a lot of fun. All right, with that, I thank everyone, and everybody have a good afternoon. Bye.